Hello, everyone. Welcome to today's presentation, and thank you for joining. My name is James Caffrey. I'm a Director of Strategic Marketing for Genomics here at QIAGEN. And the title of my talk today is Accelerating Genomic Surveillance of SARS-CoV-2 with High-throughput Next-Generation Sequencing. We've been able to do that with this new next-generation sequencing product, the QIAseq Direct SARS-CoV-2 kit, which significantly accelerates and simplifies the workflow for amplifying, enriching, and sequencing the whole viral genome. This kit is designed specifically for short-read sequencers from Illumina, and it has a number of other positive features that are a consequence of those workflow improvements, like requiring much less plastic wear compared with most existing Arctic-based SARS-CoV-2 sequencing workflows. And I'll describe those and other features of this kit a little bit later. The overall solution I will cover also includes a new data analysis and variant calling service that allows rapid scale-up of informatics capability without your lab having to invest in IT infrastructure or additional bioinformatics expertise. These data analysis and variant calling steps are also rate limiting for many labs at the moment, so we've built upon our core QIAGEN Digital Insights bioinformatics platform to address that need as well. So before I begin, let me just highlight that these QIAGEN products that I will talk about today are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. And so for up-to-date licensing information and product-specific disclaimers, please see the respective QIAGEN Kit Handbook or User Manual. QIAGEN Kit Handbooks and User Manuals are available at www.qiagen.com or can be requested from QIAGEN Technical Services or from your local distributor. So here's how I've broken out the talk today. I'd like to start with an overview of where we stand in terms particularly focused on sequencing of the genome, where we stand with respect to the pandemic, and focus on how QIAGEN has responded to those needs. Then I'll take some time to describe this new direct SARS-CoV-2 kit that we've just launched and talk about the workflow improvements I mentioned. I'll then cover some data regarding that kit, where we compare the performance of this new kit with our existing Arctic-based SARS-CoV-2 sequencing solution. And finally, I'll wrap up with a brief overview of the new service that I was mentioning for data analysis and variant interpretation using called our QIAGEN CoV-2 Insights service. So I want to start by noting that QIAGEN, like many of our peer companies, has a full spectrum of responses to the pandemic that includes both research products, which are highlighted here, some of which are highlighted here, as well as diagnostic testing of patient samples. We have a variety of solutions on the market for that, and other products over here on the right-hand side that are also grayed out, which focus on host pathogen interactions. These studies are particularly important for vaccine development and the development of therapeutics. The kit that I'm going to talk about today, the new direct SARS-CoV-2 sequencing kit, falls squarely into this area for genomic epidemiology and surveillance. And so I'm going to focus on this section here, targeted viral whole genome sequencing. That's where this kit sits. So for the rest of the talk, what I'll do is compare this new kit with our existing solution and how we've made improvements to the overall workflow. Now, the motivation for doing this is probably very understandable to every one of you who are in the public health labs around the world, or indeed any other research labs that are working on SARS-CoV-2 sequencing. Obviously, a number of variants of concern have emerged around the world in the last number of months. And indeed, they've not just emerged, they've become dominant in and fixed in various populations and regions around the world. A couple of them are shown here, the B.1.1.7 variant or lineage, which first emerged in the United Kingdom and has spread worldwide. It's now the dominant lineage here in the United States as well. B.1.351 emerged in South Africa. It's suspected to have an impact on the efficacy of some vaccines that are already out there. And new mutations continued and variants continue to emerge. For example, the P1 and the P2 variants that have sort of come out in Brazil, as well as other regional variants that continue to show up. So there's an ongoing evolution of the virus, of course, and that means that there has been a massive increase in demand for sequencing the virus to help understand which mutations are actually circulating in a population, and to identify new mutations that may emerge that could potentially become variants of concern. Now, what that has done from a consequence perspective, among other things, is it has created a very large pressure again on supply chains, for example, for labs around the world, for not just now for testing reagents, as happened at the very beginning of the pandemic around this time last year, but now for products that are needed for research as well. So we see, for example, that materials like pipette tips, plastics, plates, and other kinds of materials are in short supply for many labs. And so combined with the need to do massive sequencing scale-up, we've realized that there was a need to look at the workflow that currently exists for sequencing the SARS-CoV-2 virus and see what we could do to improve both the speed, shortening the workflow, increasing the throughput, and ideally reducing the need for lab consumables. Now, just to focus on the variants for a moment, and I'm going to highlight B117 and B351 here, just to kind of give a little bit of additional context. What you can see is that each of these lineages, as they're called, includes mutations that span across the entire viral genome. Everybody focuses, or a lot of people focus on spike protein for good reason, but you can see that these mutations occur in other open reading frames across the genome as well. And the mutations are a variety of types. This is the list of non-synonymous mutations that occur in the B117 on the left-hand side here, and in this table, the B351 lineage in the middle of the slide. And you can see that most of these mutations are point mutations at the nucleotide level, which lead to amino acid changes in the respective protein. But in addition to that, there are also deletions, as you can see here in the spike gene for B117, where there's a deletion at this position that removes a single amino acid, and actually a double deletion here, side-by-side removal of two amino acids in this case. So these mutations are particularly well studied in spike because those mutations are suspected to, and in some cases actually have been demonstrated to show increased viral transmissibility and or pathogenicity. And in fact, in the case of B117, some of these mutations have indeed been shown to be both more transmissible as well as more pathogenic than the ancestral strains that have been circulating in the first part of 2020. And not only that, there's a kind of a third factor that is really relevant now, and that is the ability of some of these mutations to potentially evade immune response. That's particularly important at this point, given the development of vaccines and the rollout of vaccines. So there's a lot of concern around the emergence of potentially new mutations that will allow the virus to evade vaccine surveillance and vaccine immune response from people. And because of all of that, really, when you want to do genomic surveillance, really the only cost-effective way of doing this in a high-throughput manner is obviously to use next-generation sequencing with a reasonably high throughput in terms of the number of samples that you can cover. So QIAGEN has actually had a solution for sequencing the viral genome out since early 2020, and that solution is it's on the right-hand side of this slide, and it's called the QIAseq SARS-CoV-2 primer panel. Now, this combination of kits that we have, a primer panel as well as a NGS library preparation kit called the QIAseq FX DNA library kit, this combination of those two kits allows for the amplification and enrichment of the entire viral genome using the Arctic-based approach. And the Arctic-based approach that I've referred to here comes from the Arctic Network, a sort of a public effort. And that team has been involved in the past in many outbreaks, particularly Ebola and others that have occurred. And very quickly after the development of the SARS-CoV-2, pandemic last year, the Arctic team developed a series of protocols and primers to enrich for the virus so that you could sequence the whole viral genome. And QIAGEN built this QIAseq SARS-CoV-2 primer panel based on that Arctic approach, which was originally developed particularly for compatibility with long-read sequencers like those from Oxford Nanopore. So the amplicons that are used to amplify the virus in the case of the Arctic approach are about 400 base pairs long. They are overlapping so that you get redundancy across the entire genome, and they give very high levels of sequence coverage and uniformity, and we see that with our primer panel as well. They also, in the case that we have, when you pair our primer panel with our QIAseq FX DNA library kit, you have the ability to multiplex samples up to 384 using the unique dual indices that come along with this FX DNA library kit. Now, having said that, we have seen and we've heard from our customers that there are some challenges with this workflow, even though it gives very, very high performance. I'll show you some of that performance data in a few slides a little bit later in the presentation. But one of the challenges that a lot of labs face with this workflow is that because the amplicons are 400 base pairs in size, they require fragmentation if you want to run those fragments and sequence them on short-read sequencers. It's also a workflow that involves ligation steps in our case, with a number of bead cleanups and quantification steps. And so what we have found is that in practice, executing this workflow for a reasonable number of samples can easily take a day and a half or more. As we looked at the growing need for speed and scale up and throughput, we realized that we needed to look again at this workflow and see how we might be able to optimize it and the entire kit and optimize it. And that's where the new kit that I'm talking about today, the QIAseq Direct SARS-CoV-2 kit, comes from. Right. This is a kit that it's a single box solution for doing both viral amplification as well as NGS library preparation. So it's just one kit. In particular, we've been able to reduce the workflow down to about a 4.5 hour library prep workflow. It requires fewer steps, dramatically less hands-on time and fewer steps. There's no fragmentation, tagmentation, or ligation involved. And that has actually allowed us to drop the amount of plasticware needed for this kit compared to our Arctic solution over here. And it still provides extremely highly uniform, in fact, even better, more highly uniform coverage of the genome than even with our primer panel. And I'll show you in the next few slides some of that data. A key component of this kit is that it uses shorter amplicons, 250 base pairs or so in size. So that's why no fragmentation is needed. So if we go now to just talking a little bit more about this kit and about the workflow itself, let's just go into that in a little bit more detail. And as this slide sort of alludes to here, the race against the virus, every minute counts. We can certainly see that in some of the very challenging news that is coming out of various regions around the world at the moment. So to kind of reiterate the three most important things to say about this kit, it includes a simpler workflow, which reduces the overall time, the workflow time, by about half when we compare it with the Arctic-based approaches. It has shorter fragments, shorter amplicons, and overlapping amplicons in two pools of primers, so it doesn't require fragmentation. We've been able to remove the ligation steps by doing PCR-directed indexing, in other words, adding unique dual indices to these amplicons using PCR rather than ligation. And because of that, and I'll show this in a little bit more detail in a moment, less plasticware required step by step. The coverage that we see with this kit is just as good, and in many cases quite significantly better than what we see with our current Arctic-based workflow, both at the uniformity level in terms of gene-specific and across the entire viral genome and coverage. It's designed for high-throughput sequencing on Illumina instruments, so these unique dual indices are particularly designed for use on Illumina sequencers. And these overlapping primer pools are obviously designed much more for short read sequencing. We have launched the product already just recently with unique dual index sets that allow multiplexing up to 384 samples at a time, and we are just about to add to that list a whole other set of UDIs that will ultimately allow you to multiplex up to 768 samples on one sequencing run. Now, obviously, that number of samples requires a NovaSeq and the right kind of flow cell. And that additional set of UDIs is coming shortly. So here's the comparison, if you will, side by side of the current Arctic workflow on the bottom of the screen here with the new shorter workflow for the for the direct SARS-CoV-2 kit. And as you can see, with the Arctic-based workflow, there are these first steps here are done with our primer panel, and then with our QIAseq FX DNA library kit to actually generate the libraries that can be sequenced on the sequencer. I should point out that our primer panel is also compatible with other library prep kits from other vendors, so you're not just restricted to using a QIAGEN kit here for the NGS library prep. This workflow, as we said earlier, takes 10 to 16 hours or so, not all hands-on, of course, but elapsed time when you've got a reasonable number of samples. What we've been able to do is remove a number of steps in that workflow, and you can see that here for the direct kit in the upper part of the slide. So you go from viral RNA sample through cDNA synthesis, direct enrichment of the viral genome. There's a B cleanup in the middle, a quantification and normalization step here, then you library index and amplify, another beam cleanup, and then your libraries are ready to go onto the sequencer. And in our hands, we can process 96 samples using manual methods, actually using multi-channel pipetter, for example, in 4 to 5 hours. We are also recognizing that this is a streamlined workflow that has a greater option for automation, and we're actively working on a number of automation scripts to automate this on some robotic liquid handlers. There'll be more information of that coming from QIAGEN shortly. So removing these workflow challenges, it's a simpler workflow, less hands-on time, and as I said, the workflow supports automation, or it will shortly when we provide some automation scripts. You can use any high-quality RNA isolate for your sample input. There's no need to dilute that RNA or to pre-qualify it. No need to do any reverse transcription primer hybridization. We've got overlapping, as I said, and redundant primer design pools that create these 250 base pair amplicons. It's a fixed number of cycles regardless of the copy number of the virus going into your front end of this workflow. Single cleanup sample here at this point. This protocol is designed to work with plates, so it can be done with multi-channel pipetters in reasonably high throughput as well. And then on quantification and normalization, we're actually looking at the moment at whether we can remove this step as well to accelerate the workflow even further. And any updates that will be published in updated versions of our kit handbook when they come out. Then you add the indexes and library amplification, and again, single cleanup per sample at the end with this protocol, obviously, as I said, designed to work with plates. Now, let me just move to some data on the performance of this kit. So this slide shows in the upper two traces the library yield that we get from the QIAseq Direct SARS-CoV-2 kit. And in the lower two traces, examples of library yield that we get from our Arctic-based primer panel approach. And what you can see in the primer panel down here, just to start there, is you can see that there's a so-called sort of a hump and a peak for whether you're doing 100 copies or 1,000 copies input into this library preparation. And that hump and peak represents the fact that you've got amplicons that are about 400 base pairs in size with some extra tags on either end that then have to get fragmented in order for them to be run effectively on a short read sequencer. And so the hump that you see here is the fact that it's representative of the fact that that fragmentation does not necessarily always go to completion. Now, even though you've got a hump and a peak, we find that and our customers find that they still get excellent genomic coverage, both from a coverage perspective and a uniformity perspective with our primer panel. When you run the same kind of analysis with our direct kit, what you can see is a trace that's sort of much more similar to what people expect to see from a library yield. A much tighter peak, again, irrespective of the number of copies going in at the front end. And I would just draw your attention to the fact that the scale on the x-axis here on the y-axis for the upper traces is significantly higher than for the lower ones. So you also get a much higher yield of library using the direct SARS-CoV-2 kit. Now, when we look at performance from a uniformity and a coverage perspective, we find that with our Arctic-based primer panel approach, which is shown in the red bars here, we get excellent coverage at 1x of the mean, almost 90% at 1x of the mean. That does drop a bit when you plot these data as 0.2x of the mean. What we see with our direct kit, which is shown in blue, the two blue bars here, is that we're getting 99+% coverage at 0.1x of the mean, and in the mid-90s for 0.2x of the mean. So we see an increased level of uniformity with our direct kit compared to even with our Arctic primer panel. And when we look at base level coverage depth at different depths, you can see that again for the Arctic-based primer panel in red, we've got pretty good coverage, base level coverage at 20x sequencing depth, almost 99%, but that does drop down to about 95, 96 at 100x coverage. We see no such equivalent drop for the direct kit. So all in all, we think these performance numbers, and these are, I should point out, are done with ATCC positive controls. We see excellent performance and even better performance with the direct kit, even compared with our already good Arctic-based approach. What does that coverage look like from a gene-specific perspective? So these three traces here just dig into the S gene, the N gene, and the endoribonuclease gene. And the red trace is the coverage uniformity for the Chi-seq SARS-CoV-2 primer panel, in other words, the Arctic approach. The blue trace is the coverage for our new direct kit. And what you can see with the Arctic approach is that, again, these longer amplicons, you can see that there are areas, it's already pretty good coverage, 1000x, but there are definitely areas of peak and trough in this red trace, whereas the direct kit provides a much more uniform coverage across the length of the spike gene here with the direct kit. There are some areas of higher and lower, but it's generally much more uniform than what we see with our Arctic approach. Same thing is true for the N gene, and likewise for the endoribonuclease gene. And this is particularly worth pointing out that for endoribonuclease gene, we did see early on that there were regions of this gene that sort of dropped out. And even with our Arctic approach, we had to add additional primers across this region and rebalance the concentrations of some of the primers to get this level of coverage that you see in the red trace. Again, when you look at that equivalent approach with the direct kit, the new direct kit in blue, you see just a much more uniform level of coverage and indeed higher levels of coverage. Now, the graph is an attempt to, in a static format, to show what that coverage looks like across the entire genome. So we're looking at here from base 1 to base almost 30,000 of the entire SARS-CoV-2 viral genome. And again, what you can see in red, our Arctic-based approach, in blue, our direct kit approach. There's more jaggedness to these traces, of course, because we're looking at an entire viral genome in one slide. But I think it's pretty obvious that to just even by eye that both the coverage is higher with our direct kit in blue compared to our Arctic-based approach, and the uniformity is broader. The only two regions I would point out where we don't have good coverage are right at the very 5' end of the genome, the first 50 or 60 bases or so, and right at the very 3' end of the genome. And that's true given the fact that this is a amplicon-based tiling approach to amplify this virus, and there's just no other way of getting those regions with that kind of approach. Okay, I'm going to wrap up with just some short words on our data analysis and variant calling solution from our digital insights team here. We have launched a new QIAGEN CoV-2 Insights service, and this basically is a managed service that we provide to labs where we will essentially take FASTQ files from that have been generated off a sequencer, analyze them at scale in the cloud, provide a report, an example of which is shown here on the right-hand side of this slide, with a variety of pieces of information, which I'll go into in a bit more detail now, and then hand you back that report. And so you can sort of scale your analysis of data through the web, essentially, without having to invest in IT infrastructure or indeed additional bioinformatics expertise or resources. This service supports most of the SARS-CoV-2 sequencing panel options that are out there, so not just QIAseq ones, the primer panel and the direct kit, but others as well. It's fast and accurate and secure, so wherever you are in the world, this is a solution that you can use scalably. No need for infrastructure and expert resources. It's fully automated, and we provide lots of quality control steps. It's easy to integrate the results you get back from us with your existing research IT infrastructure, and so on. And the data that we send back is capable of being analyzed in our workhorse data analysis solution for NGS, the QIAGEN CLC Genomics Workbench. And we have a pricing model here based on sample usage. So here's what that report looks like. You can tag it to your own, you can brand it and logo it for your own lab if you want. It provides a lot of QC metrics very visibly around the quality of your run and the data. The data analysis is performed, the workflow name and version number and so on are provided. And then the report also includes information about the reference sequences that were used for the workflow, lineage predictions by pangolin and clade predictions from NextClade. And of course, most importantly, probably at the moment, coverage and variant profiles for the genes that are covered, divided into consensus variants and lower frequency variants in a table at the bottom of the report. This is what the contextual perspective. Your lab is here. You provide FASTQ files. We do all of the work in the cloud, send you back outputs that you can then incorporate into your own LIMS system or other research IT infrastructure that you have. You can look at a lot of this data in the CLC Genomics Workbench, and for each sample, a number of different types of reports are provided. So with that, let me summarize and just come back now to the kit for a moment and say for our QIAseq Direct SARS-CoV-2 kit, it comes in two kit sizes, 96 samples and 384 samples. For the 96 sample configurations, we have at launch now 4 kits. They follow the same kind of common nomenclature for indexed kits, kits A, B, C, and D. Each of these has 96 UDIs in them, and they can all be mixed together to get to 384 multiplexing in one go. And as I said earlier, we are going to be launching another set of UDIs. This will be kits sets E through H, and they can also be multiplexed with the A through D kits, and so you now have the ability to scale up to 768 samples at a time. And with that, I would like to thank you for your attention today. If you have any questions about this, please contact us at QIAwebinars@QIAGEN.com. Thanks again for your attention. I hope you have found this useful, and have a great rest of your day.